Yeah, you can start now.
Yes, sir. We can start now.
Good morning, sir. Good morning to all of you. I would like to introduce myself first.
Sir, start.
Prabhakar. Shall I start?
You carry on. Start.
Yeah, good morning to all of you. I would like to introduce myself. This is Kashi Vishwanath Chinta, Organizing Secretary of IPGA-Telangana State, and the chairman for today's webinar. I welcome to national webinar to all distinguished guests and attendees. I invite Dr. Nasaji for this webinar and deliver his speech on today's webinar. I also welcome Sri Kodeshwara Garu, President IPGA, Telangana State, and Speaker Dr. Prabhakar Reddy and Co-Chairman Vijay Kumar. Sir, Dr. Nasaji, I request you to start your request you to deliver your speech, sir.
Thank you very much, and very good morning to all of you. At the outset, my heartiest congratulations to IPGA Telangana branch for organizing this webinar on role of nanotechnology in personalized medicines. Very respected Shri G. Koteshwar Rao, the President of IPGA Telangana branch, Shri Kashi Vishwanath ji, the Organizing Secretary cum Chairman of the today's webinar, Dr. Gampa Vijay Kumar, the Professor of Pharmacy, Speaker of the Day, Dr. V. Prabhakar Reddy, who is the head of UCPS Mehboob Nagar and the Secretary of IPGA Telangana State, other professionals, participants, and delegates who are attending this webinar. The topic for this webinar, that is role of nanotechnology in personalized medicine. This is the need of the day. We know that entire world is suffering from lifestyle disease. And one of the disease is the cancer. So what I understand that personalized medicines are based on each patient's unique genetic makeup. These medicines can be used in the treatment of cardiovascular diseases, HIV infections, arthritis, lungs disorders, and so on. Nanomedicines are nowadays defined as nanoscale tools for diagnosis, prevention, and treatment of disease. Nanomedicines are also reported as nanodrugs, nano-carriers, nanoparticles, nanomaterials, and nanotherapy. If I understand, the nanotechnology was developed long back in 1990, and about in 1995, first FDA drug which was approved in the treatment of cancer was Doxil, which was used in the treatment of breast cancer and ovarian cancer. Nanomedicines are also used in the treatment of rare diseases. We know that pharmaceutical industry is mainly concerned with the treatment of the other diseases, but there is few development molecules for the treatment of rare disease where the nanomedicines has to play a very important role. Even tuberculosis. Now, if we see the tuberculosis, in the treatment of tuberculosis, we are taking the repetitive medicines for a longer period of time. But nanomedicines play an important role in reduction of drug dosages and decrease in the frequency of administration of drugs. With these remarks, I welcome this today's speaker, Dr. Prabhakar Reddy, and he will be presenting a detailed presentation on role of nanotechnology on personalized medicines. So my best wishes to the IPGA Telangana State Branch and to all the participants. Thank you very much.
Thank you, sir. Thank you very much for your valuable words, sir. Thank you very much. Now may I request our President, Sri Kodeshwara Garu, to deliver your speech.
Yeah. Respected Dr. Atul Nasaji, the President of IPGA National, and today's chairman, Sri C.H. Kashi Vishwanathji, organizing secretary, IPGA-Telangana, Dr. Gampa Vijay Kumar, co-chairman of this today's event and treasurer of IPGA-Telangana, and today's speaker, Dr. V. Prabhakar Reddy, General Secretary, IPGA-Telangana, and my friend, Dr. M.C. Nishit from Kerala, and a special welcome to Miss, maybe, I don't know what to call, Vaishali Veera Reddy, daughter of Dr. Prabhakar Reddy, who is also witnessing this. It is a great and proud moment for him, who is also a pharmacist, postgraduate pharmacist. A hearty welcome to Indian Pharmacy Graduates Association, Telangana, and the dignitaries to today's webinar on role of nanotechnology in personalized medicine. Friends, Indian Pharmacy Graduates Association, the only exclusive pharmacy professional group in India, has been contributing to the growth and development of careers in pharmacy throughout its various branches such as Students Forum, Women's Forum, Industry Forum, Regulatory Forum, and the Teachers Forum by conducting technical seminars, awareness activities, and other social activities with the help of IPGA Welfare Trust. The association has been constantly engaged in conducting workshops, annual conferences, seminars, and other activities to update the knowledge and skills of pharmacy professionals. IPGA was established in the year 1973, registered under Societies Registration Act 1860. This association is affiliated to Indian Pharmaceutical Congress Association, IPCA, the federal body of the five associations, namely IPGA, IPA, IHPA, APTA, and AIDCOR. The association has been working for past 40 years for the upliftment of professional of pharmacy. It has a mission to improve the professional status of pharmacy graduates and to secure their rightfulness place in pharmacy and allied professionals. At present, the association has more than 9,000 live members in about 21 state branches all across the country. Our mission and values are in order to provide strength to the profession of pharmacy. We feel that the pharmacy graduates from all corners of the country should join hands under the banner of IPGA to provide inputs on the national issues and programs. Coming to the today's program on continuous educational programs and upgradation of the pharmacy profession. Every month we are conducting a webinar on a selected topics, and I am very sorry all these days I could not able to recognize my friends, my members who are the number one technocrats and highly knowledgeable professors are there in my group, and I starting with Dr. V. Prabhakar Reddy Garu, the professor, and our General Secretary IPGA, who is also recently elected, nominated as Pharmacy Council of India Central Council member, and we congratulate him. And I hope this event will be useful to the teaching faculty, enforcement faculty, and students fraternity. And I thanks to this time really I have not interfered with this event, especially because I am engaged with some other forthcoming event. So I especially thanks to Sri Kasi Vishwanath, Dr. V. Prabhakar Reddy, Dr. Gampa Vijay Kumar, and Dr. Nishit for arranging this in a successful way. I wish all you the best, and over to the Dr. Kasi.
Thank you, sir. Thank you, President Ji. It's a very pleasure to have you here on this day. Thank you very much for your valuable suggestions. Thank you, sir. I now request our Dr. Gampa Vijay Kumar to introduce our today's speaker, Dr. V. Prabhakar Reddy.
Good morning, all. It is my pleasure to introduce Professor Veera Reddy Prabhakar Reddy, who is an active member in pharmacy field and secretary for IPGA Telangana State, and then recently nominated as Central Council member, Pharmacy Council of India. He done his B Pharmacy from Kakatiya University, Warangal, PG from Andhra University, PhD from Kakatiya University, PDF from Butler University, and presently he is working with Palamur University as head since 2013. Particularly, previously he's worked with the St. Peter's College of Pharmacy, Chaitanya College of Pharmacy, and then NNR College of Pharmacy. And then presently, his research work is presented in national and international conferences of FIP, like China, Egypt, Lebanon, Turkey, Thailand, and also in India. He is also a recipient of awards and fellowships like Association of Biotechnology and Pharmacy, Rashtriya Vidya Saraswati Puraskar, Broomfield Foundation Award in FIP. He also authored a number of the textbooks and then reviewer for more than 100 national and international journals. Under his guidance, many renowned scholars were awarded with the doctoral degree. I request Prabhakar Reddy Garu to deliver today's talk.
Thank you, Vijay. Now, I'm sharing my talk of today. Is it visible to all of you?
Yes, visible.
Yeah. First, I will thanks to our national president, Atul Nasaji, for giving a lot of motivation to conduct continuous medical education programs since 2 years. Recently, our Telangana branch started 2 years back only. Now, special thanks to our president, Kodeshwara Garu. Always he will look for the education concept. Then my special thanks to Organizing Secretary, Kashi Vishwanath Garu, and Treasurer, Gampa Vijay Kumar Garu, and special thanks to Nisit, who is giving and supporting this webinar. Okay, so my topic of today is role of nanotechnology in personalized medicine. So last 20 years, I'm in this field, nanotechnology. Lot of changes are coming. When I started this work, only 10,000 abstracts are there. Now it went to 1 lakh plus. So that means a lot of research is going on. But unfortunately, only 10% is coming from industry. 90% is academia. So that is the reason. So we can expect more from industry. Then only it will reach to the common public. So today my objectives are drug delivery, nanotechnology literature review. What is nanotechnology? What is targeting? Drug delivery carriers, drugs and biologically active materials. What is personalized medicine? Applications, issues, challenges, and conclusion. So I'm giving a little bit basics because many of students are there in this webinar. So I'll come to starting. Slowly I will take up to nanotechnology, then personalized medicine, then finally come to conclusion. So it will 60 minutes minimum, so 90 minutes maximum. So please bear. And any doubts now you can keep in the chat box. End of the talk, I'll give the answers. Or you can touch into my mail ID also, I'll give the answers. So you know the definition of drug delivery, the appropriate administration of drugs through various routes in the body for the purpose of improving health is called drug delivery. It is highly interdisciplinary. It is not a young field. It has recently evolved to take into consideration drug physiochemical properties, body effects and interactions, improvement of drug effect, patient comfort and well-being. This is very, very important. Then only the products will move into the market. So this comes under controlled drug delivery. So drug delivery you can broadly classify into conventional and
Excuse me, Prabhakar sir.
Yeah, please.
Sir, your slides are not moving.
Not moving?
Not moving. Sir, can you make it presentation mode?
Okay, 1 minute. So once again, I'm sharing. Is it okay, sir?
Yes, now.
Yes, yes.
So drug delivery is two types, conventional and control. So conventional, you can see enteral, parenteral, and control, sustained, extended, site specific, and pulsatile also. Okay. So coming to oral administration, so convenience, not invasive, higher compliance, and manufacturing is well established, and a lot of infrastructure is there, but few disadvantages. We cannot give to unconscious patients, low solubility, low permeability, because most of the drugs are insoluble in nature. So there we are getting little problems. The degradation by G enzymes, first-pass metabolism, food interactions, and irregular absorption. So traditionally oral delivery systems like tablets, capsules, soft gelatin capsules, suspensions, emulsions, elixirs. Coming to buccal or sublingual, bypass first-pass metabolism, rapid absorption, low enzymatic activity, and little disadvantage like discomfort during dissolution, swallowing or loss of effect, and small doses. The main drawback here is we cannot go for higher doses. Like tablets and chewing gums are available in the market. Coming to rectal, so advantages, bypass first-pass metabolism, useful for children, and disadvantages like absorption depends on the disease state, degradation by bacterial flora, and uncomfortable, especially the kids not happy with this drug delivery. So generally we have suppositories and enema. Coming to intravenous, 100% bioavailability, because here there's no wastage of the drug, directly goes to systemic circulation. Rapid response, total control of blood concentration, maximize incorporation of degradable drugs, bypass first-pass metabolism, and disadvantages invasive, because it's a pain causing. Trained personnel are required. We cannot take like oral. Possible toxicity due to incorrect dosing, and sterilization is very, very important because they're going directly into the systemic circulation. So generally you can see injections like bolus, IV bag, infusions. Coming to subcutaneous advantages, patient self-administration, slow, complete absorption, bypass first-pass metabolism, and disadvantages, invasive, irritation, inflammation. Maximum dose is only 2 ml. So here dose, we cannot go more than that. Coming to intramuscular advantages, patient can administer the drug himself, larger volume than subcutaneous, bypass first-pass metabolism. But disadvantages, invasive, patient discomfort, irritation, inflammation, may require some training. Coming to inhalers, bypass first-pass metabolism, gases are rapidly absorbed. Disadvantages, solids and liquids can be absorbed if size is below 0.5 micrometers. Coming to transdermal, advantages, local effect, ease of administration. Just you can keep wherever you want on the body surface. Here the disadvantage, low absorption for some drugs, may cause allergic reaction. And coming to limitations, the dosage should be less than 10 mg per ml and molecular weight is less than 1000. So factors, drug physicochemical properties like molecular size, half-life, chemical stability, denaturation, degradation, and solubility is a big issue. You know, BCS, that is biopharmaceutical classification system. According to BCS, class 1, class 2, class 3, class 4. Class 1 is high solubility, high permeability. Class 2, low solubility, high permeability. Class 3, high solubility, low permeability. And class 4, low solubility, low permeability. So 2 and 4 are the problematic. And we should think about the pH, pK, temperature, concentration, crystallinity, particle size, and state of hydration. And drug biological interactions, sensitive to first-pass metabolism, efflux pumps, hydrophilicity, high density charge, enzymatic degradation, bacterial degradation, half-life, and side effects. So these are all factors influencing the drug absorption. So coming to delivery route, so desired pharmacological effect will come either local or systemic or immediate response, and dose size and molecular size is very, very important. And these two terms are very, very important in our pharmaceutics, pharmacokinetics, pharmacodynamics. So in the kinetics, we should think about the dosage regimen, where, how much, how often, how long. Coming to dynamics, plasma refers to the clear supernatant fluid that results from blood after the cellular components have been removed. So this graph is very, very important, right from your B Pharm, M Pharm, PhD, postdoc, lifelong. Time versus concentration. This is plasma concentration. See, there are 3 lines: subtherapeutic, therapeutic, toxic. So any dose, as per regimen, if you are not taking, it comes under subtherapeutic. Therapeutic, exact dosage regimen, either 2 or 3 tablets as per the requirement. Then it should be therapeutic window. And if you take, instead of 1 tablet, if you take 3 tablets at a time, it goes to toxicity. That means adverse effects. So that is the reason no, we should not take overdose or underdose, because we are the pharmacists. We should follow the dosage regimen. Then only the effect is impact on our body. Okay, so this table, try to remember lifelong. And you see another graph, time versus plasma concentration, successful therapy, unsuccessful therapy. For example, if you take Dolo 650, daily 3 tablets. So what? Every 8 hours you should take. Okay, every 8 hours if you are taking, good. It comes under 3 into 8, 24 successful therapy. So instead of taking 3 tablets, if you take 1 time or 1 missing, so that means unsuccessful therapy. There your disease will be again comes to back. So this is about drug delivery. Now I am coming to nanotechnology. You see, I told you the nanotechnology. If you go to PubMed, just type nanotechnology, you are getting 1,26,402 abstracts from 1978 to 2022. Yesterday I did it. When I started my research in 2000, that time only 10,000 came. So that means a lot of research is going on. And from that, 90% is coming from academia. I already told, only 10% is from industry. And people are saying that 100 nanometers is the minimum now. When I did my research, that 1,000 they said, but now they're coming to 100 nanometers. And they're given a lot of definitions by FDA. If you go to Google, you get a lot of definitions. Here I'm giving very important definitions. Nanotechnology is the understanding and control of matter at the dimensions of roughly 1 to 100 nanometers. The method by which a drug is delivered can have a significant effect on its efficacy. Some drugs have an optimum concentration range within which maximum benefit is derived. And concentrations above or below this range can be toxic or produce no therapeutic benefit at all. On the other hand, the very slow progress in the efficacy of the treatment of severe diseases has suggested a growing need for a multidisciplinary approach to the delivery of therapeutics to targeting tissues. So why nanotechnology came into picture? Because we are giving a lot of doses. The more dose, what happens to more side effects. So in the nanotechnology, we are reducing the dose and we are targeting to the specific site. That is the main concept of this nanotechnology. So from this, new ideas on controlling the pharmacokinetics, pharmacodynamics, non-specific toxicity, immunogenicity, biorecognition, and efficacy of drugs were generated. These new strategies, often called novel drug delivery systems, are based on interdisciplinary approaches that combine polymer science, pharmaceutics, bioconjugate chemistry, and molecular biology. To minimize drug degradation and loss, to prevent harmful side effects, and to increase drug bioavailability and the fraction of the drug accumulated in the required zone, various drug delivery and drug targeting systems are currently under development. So here I'm saying bioavailability. What's the bioavailability? Rate and extent of the drug available to systemic circulation. So that is the reason today nanotechnology has a lot of demand in the market. Among drug carriers, one can name soluble polymers, microparticles made up of insoluble or biodegradable natural and synthetic polymers, microcapsules, cells, cell ghosts, lipoproteins, liposomes, and nanoparticles. The carriers can be made slowly degradable, stimuli reactive, and even targeted. Coming to nanotechnology definitions. Again, you will get a lot of definitions in the literature, but I'm giving only a few. the development and use of devices that have a size of only a few nanometers. Branch of engineering that deals with things smaller than 100 nanometers. Nanotechnology, sometimes called molecular manufacturing, is a branch of engineering. The art of manipulating materials on an atomic or molecular scale, especially to build microscopic devices. See here I'm giving what is tetra, tera, giga, mega, kilo, milli, micro, nano. So in the nano, you see 10 power of minus 9. Giga, you see 10 power of 9. Mega 10 power of 6. Here, micro 10 power of minus 6. Kilo 10 power of 3. Milli 10 power. So we are coming down reducing the size of the particles. That is the beauty of here. So here they're given some recommendations. So they are asking minimum 220 nanometers is required. So coming to nanotechnology. So a nanometer is a unit of length in the metric system equal to 1 billionth of a meter. That is called 10 to the power of minus 9. Technology is the making, usage, and knowledge of tools, machines, and techniques in order to solve a problem or perform a specific function. So that is the full definition of nanotechnology. So nanotechnology is the study of manipulating matter on an atomic scale. Nanotechnology refers to the constructing and engineering of the functional system at very micro level, or we can say at atom level. A nanometer is 1 billionth of a meter, roughly width of 3 or 4 atoms. The average human hair is about 25,000 nanometers wide. Imagine how small a nanometer. And we should thankful to this guy. The first ever concept was presented in 1959 by the famous professor of physics, Dr. Richard P. Feynman. Invention of the scanning tunneling microscope in 1981 and the discovery of fullerene, that is carbon-60, in 1985 lead to the emergence. The term nanotechnology had been coined by Norio Taniguchi in 1974. The early 2000s also saw the beginning of commercial applications of nanotechnology, although these were limited to bulk application of nanomaterials. Silver nano platform for using silver nanoparticles as an antibacterial agent, nanoparticle-based transparent sunscreens, and carbon nanotubes for stain-resistant textiles. So tools and technology. There are several important modern developments. The atomic force microscope, AFM, scanning tunneling microscope, STM, nanolithography, such as optical lithography, X-ray lithography, dip and nanolithography, electron beam lithography. So what is lithography? In MEMS context, is typically the transfer of a pattern into a photosensitive material by selective exposure to a radiation source such as light. And you may ask, what is the equipment required for this nanoparticles? We need homogenizer, ultrasonicator, mills, spray milling, supercritical fluid technology, electrospray, ultracentrifugation, nanofiltration, and we need digitizer. But unfortunately, if you want to establish a nanotechnology, you need at least 50 lakhs to 1 crore. Then only we can do this type of research. That is the reason many universities are not affording this technology because of lack of cost. Coming to nanoparticles for drug delivery. So there are different types: metal-based nanoparticles, lipid-based nanoparticles, polymer-based nanoparticles, biological nanoparticles. So nanobiopharmaceuticals. Main technologies covered: liposomal, monoclonal antibody-based, polymer-based technologies, host of newer technologies such as nanoparticles, including various nano-dimensional entities such as molecular imprinted polymer, metallofluorines, prodrug delivery, oral, injectable, and implantable, pulmonary, and transdermal and transmucosal delivery have come up. Some significant achievements of nano devices: development of 1 dose a day ciprofloxacin using nanotechnology, human targeted taxol delivery using nanoparticles, improved ophthalmic delivery formulation using smart hydrogel nanoparticles, oral insulin formulations using nanoparticle carriers, liposomal based amphotericin B formulation. These are few examples which are the technology used by scientists. Coming to priority areas, so nanotechnology cannot go for all areas. Main, cancer technology, cancer nanotechnology, like diagnosis, human target delivery, imaging, and cancer gene therapy. And there are different types of there. Nanopowders are there, nanoclusters are there, nanocrystals are there, polymeric nanoparticles are there, carbon-60, carbon nanotubes. Because no, these are not related to our talk, so I'm skipping. Nano rods are there, nano boards are there. Coming to approaches. So two types of approaches, bottoms up. So in the bottom approach, different materials and devices are constructed from molecular components of their own. They chemically assemble themselves by recognizing the molecules of their own breed. Second, top-down approach. In top-down approach, nano objects and materials are created by large entities without bonusing its atomic reactions. Usually, top-down approach is practiced less as compared to bottom-up approach. Solid-state techniques can also be used to create devices known as nano electromechanical systems, NEMS, and micro electrochemical systems, MEMS. So you should remember this terminology, NEMS, MEMS. MEMS became practical once they could be fabricated using modified semiconductor device fabrication technologies normally used to make electronics. So generally, what type of metals are using? Zinc oxide, silver ion, aluminum silicate, gold ions. And you see the importance of nanotechnology applications. You see, only we can see 25% in the pharma, but 75% in other technology like energy, optical engineering, defense and security, bioengineering, cosmetics, nano fabrics, nano devices, nano biotechnology, medicine and drugs. So my interest is on medicine and drugs. Coming to nanotechnology, cancer drugs provide new options for drug delivery and drug therapies, enable drugs to be delivered to precisely the right location in the body, and release the drug doses on a predetermined schedule for optimal treatment. See here what happens if you take generally cancer comes, what happens? We are using three concepts, either chemotherapy, surgery, or radiation. So which is first, which is last will decide by the doctor. So the minute the person is getting cancer, okay, surgery is done, again is coming. Radiation done, again is coming. Third option, doctors will go for chemotherapy. The present chemotherapy drugs, unfortunately, they will go to all over the body and destroy the good cells. At the same time, they will kill the cancer cells, but other good cells also dying. Because of that, patient is dying, not because of cancer, because of chemotherapy. So there are, when cancer comes, we got the same stage 1, stage 2, stage. If it's stage 1, I can say 75% survival rate. Stage 2, 50%. Stage 3, only 25%. But unfortunately, these cancers are coming, are knowing to for us at the third stage. So that time, doctors doesn't have any option. They cannot go for surgery or radiation. Why? Because the cancer cells spread all over the body. So only option, they will go for chemotherapy. So that chemotherapy, again, 6 times they will give, 6 cycles they will say, every 21 days. But unfortunately, a lot of side effects. So that is a big risk. So that is the reason today the nano is coming into picture. So provide a new option for drug delivery and drug therapies, enable drugs to be delivered to precisely right location, attach the drug to nano-sized carrier because they are using carriers. They become localized at the disease site, that is cancer tumor. That is very, very important. Then they release the medicine that kills the tumor. It will not disturb other good, healthy cells. Current treatments through radiotherapy or chemotherapy, nanobots can clear the blockage in arteries. I told you, they were using in the fabrics also. In the mobiles, we are using nanotechnology. Electronics, we are using. Computers, we are using. Previously, you see first generation computer, it is the size of a big room. Today, we are seeing laptops. And coming days, you can see, like in the watch, you can see, because day by day, the size is reducing. Even TV is also you observe. Now, wall mounted TVs. Previously, you can say a big size. So because of this nanotechnology, the new concept is coming here. And other uses, cutting tools made up of nanocrystal materials such as tungsten carbide, tantalum carbide, and titanium carbide are more wear and erosion resistant and last longer than their conventional counterparts. Silver nanocrystals have been embedded in bandages to kill bacteria and prevent infection. Nanoparticulate based synthetic bone formed by manipulating calcium and phosphate at the molecular level. Aerogel's latest known solid due to good insulating property is used in the space suits and are proposed to use in spacecraft. Coming to nanotechnology in India. IIT Mumbai is the premier organization in the field of nanotechnology. Research in the field of health, environment, medicines are still on. Starting in 2001, the government of India launched Nanoscience and Technology Initiative, NSTI. Then in 2007, Nanoscience and Technology Mission 2007 was initiated with an allocation of rupees 1,000 crores for a period of 5 years. The main objective of this nano mission are basic research promotion, infrastructure development, development of nanotechnology, human resource development. So what are the possibilities for the future? Nanotechnology may make it possible to manufacture lighter, stronger, and programmable materials that require less energy to produce than the conventional material and that promote greater fuel efficiency in land transportation, ships, aircraft, and space vehicles. The future of nanotechnology could very well include the use of nanorobotics. The nanorobots have the potential to take on human tasks as well as tasks that humans could never complete. The rebuilding of the depleted ozone layer could potentially be able to be performed. There would be an entire nanosurgical field to help cure everything from natural aging to diabetes to bone spurs. There would be almost nothing that couldn't be repaired with the introduction of nanosurgery. But here, every delivery, there are some limitations, or I can say pitfalls. Nanoparticles can get into body through the skin, lungs, and digestive system, thus creating free radicals that can cause cell damage. Once nanoparticles are in bloodstream, they will be able to cross the blood-brain barrier. The most dangerous nanoparticle application used for military purpose is the nanobomb that contain engineered self-multiplying deadly viruses that can continue to wipe out a community, a country, or even a civilization. Nanobots, because of their replicating behavior, can be big threat for grey goo. Coming to targeting, I don't know. So nano targeting. Targeting is the ability to direct the drug-loaded system to the site of interest. You want to target brain or kidney or liver. So depending on your interest, you can do the formulation. Two major mechanisms can be distinguished for addressing the desired sites of drug release: passive and active. What is passive? Higher concentration, lower concentration. What active? You should give some energy. An example of passive targeting is the preferential accumulation of chemotherapeutic agents in solid tumors as a result of the enhanced vascular permeability of tumor tissues compared with healthy tissue. A strategy that could allow active targeting involves the surface functionalization of drug carriers with ligands that are selectively recognized by receptors on the surface of the cells of the interest. I will give one example. If the person got cancer, so there what happened? No, folate receptors will be overexpressed. For that, you can identify. So since ligand receptor interactions can be highly selective, this could allow a more precise targeting of the site of interest. So the minute is excess of folate, no, these nanoparticles will go there and attach to these folate receptors. Automatically, the cancer cells will die. Controlled release and subsequent biodegradation are important for developing successful formulations. Potential release mechanisms involve absorption of surface-bound adsorbed drugs, diffusion through the carrier matrix, diffusion through the carrier wall, carrier matrix erosion, and a combined erosion or diffusion process. These are all the mechanisms. For over 20 years, researchers have appreciated the potential benefits of nanotechnology in providing vast improvements in drug delivery and drug targeting. Improving delivery techniques that minimize toxicity, the very, very important one, and improve efficacy offers great potential benefits to patients and opens up new markets for pharmaceutical and drug delivery companies. Other approaches to drug delivery are focused on crossing particular physical barriers, such as the blood-brain barrier, in order to better target the drug and improve its effectiveness, or on finding alternative and acceptable routes for the delivery of protein drugs other than via the gastrointestinal tract, where the degradation occurs. Coming to carriers, I told you, so we need carriers. So colloidal drug carrier systems such as micellar solutions, vesicle and liquid crystal dispersions, as well as nanoparticle dispersions consisting of small particles of 10 to 400 nanometer diameter, show great promise as drug delivery systems. When developing these formulations, the goal is to obtain systems with optimized drug loading and release properties, long shelf life, and low toxicity. 4 important points. Optimization of drug loading means how much the drug is entering into the carrier. And okay, the drug is entered, whether it is releasing or not, we should do 24-hour study. Then how many years it is doing, that is shelf life. And we should think about the toxicity of your carrier. That is also very, very important. The incorporated drug participates in the microstructure of system and may even influence it due to molecular interactions, especially if the drug possesses amphiphilic and lipophilic properties, because the drug may be water loving or water hating. So depending on the requirement, you can make. See here I'm showing some carriers like micelles, vesicles, multifunctional dendritic polymers, liquid crystals, nano capsules, and nano spheres. These are the different carriers. There are many technological challenges to be met in developing the following techniques. Nano drug delivery systems that deliver large but highly localized quantities of drugs to specific areas to be released in controlled ways. Controlled release profiles, especially for sensitive drugs, materials for nanoparticles that are biocompatible and biodegradable is very, very important, because it should go to body. What happens? It should be compatible with your body parts and should be degradable from the body. Architectures, structures such as biomimetic polymers, nanotubes, and technologies for self-assembly. Functions like active drug targeting, on-command delivery, intelligent drug delivery devices, bioresponsive triggered systems, self-regulated drug delivery systems, system interacting with body, smart delivery, virus-like systems for intracellular delivery, nanoparticles to improve devices such as implantable devices, nanochips for nanoparticle release or multi-reservoir drug delivery chips, nanoparticles for tissue engineering, example for the delivery of cytokines to control cellular growth and differentiation and stimulate regeneration, or for coating implants with nanoparticles in biodegradable polymer layers for sustained release. Advanced polymeric carriers for the delivery of therapeutic peptides or proteins. In the development of combined therapy and medical imaging, nanoparticles for diagnosis and manipulation during surgery. Universal formulation schemes that can be used as intravenous, intramuscular, perioral drugs. Cell and gene targeting system. User-friendly lab and a chip devices for point of care and disease prevention and control at home. Devices for detecting changes in magnetic or physical properties after specific binding of ligands on paramagnetic nanoparticles that can correlate with the amount of ligand. Coming to research, focus on the development of innovative drug delivery systems for the treatment of severe chronic diseases such as cancer, leishmaniasis, ophthalmic and immunological disorders. Focus on drug targeting and enhanced oral and ocular bioavailability of poorly absorbed lipophilic drugs. So now, what is the advantage, you may ask? Solubilization of large dose of lipophilic drugs, better penetration through membranes and higher bioavailability, degradation of production in the GIT, convenient for parenteral, topical, ocular, oral, potential for drug sustained release, stable and can be sterilized, and low cost manufacturing. So these are the advantages of nanotechnology. Coming to therapeutic fields, most lipophilic drugs should be benefit from the nanotechnology platform. So here why I'm saying this one, lipophilic means what? The drugs which are insoluble in water, they are directly suitable. For example, if you see diclofenac, in the tablet form, what you are doing? Diclofenac sodium, ciprofloxacin, hydrochloride, enlapil, maleate. That means we are making into salt forms. Why we are making salt forms? To dissolve in the water. But in the nanotechnology, you cannot do any salt form. Directly you can take pure drug, and especially useful for the lipophilic character. So coming to advances, any application of science that deals with the elements between 100 nanometer and a tenth of a nanometer in size, in which size is critical to the application's ultimate purpose. Up to 50% of existing drugs and drugs in the pipeline are classified as poorly water-soluble drugs, I already told, insoluble in water. So the minute the drug is insoluble, you cannot expect any absorption, any dissolution. Automatically, it will not go to the bloodstream. So that is called bioavailability. So bioavailability is very, very poor. That is the reason, if you take nanotechnology, we can increase the solubility, we can increase the absorption, we can increase, it will go to the body, and automatically it will in the bloodstream, the bioavailability will be high. So what are we looking for in nanocarriers? It should be safe, I already told you, non-toxic, non-immunogenic, non-inflammatory, biodegradable and biocompatible. Degradation products readily eliminated from the body are used by the body. Ideally, excipients have GRAS designation by the FDA. The GRAS means generally regarded as safe. Compatible with drug, because we are mixing with the drug. So it should be compatible. Compatible with cells and tissues, because we are taking inside the body. No platelet aggregation, no RBC lysis, no neutrophil activation. Manufacturing ease and low cost, and finally that it should be stable. So nanotechnology for the targeted delivery is the right drug to the right place at the right time. So this is the concept of nanotechnology. I'm showing some examples how the liposomes looks, how the lipid nanospheres. So both are phospholipid bilayers, and you see the left side aqueous phase. That means liposomes are suitable for water-soluble drugs. You see the right side nanospheres. You see lipophilics. That means water hating. So you can go for those drugs which are not soluble in water. You can prefer nanospheres. I am showing some other drugs. You see here drug encapsulation in liposomes. You see here. So here your drug is here. These are the pegs. Here we are using anchors. These are called ligands. So this ligand will directly take to the particular area, whether you want to target liver, kidney, brain. So these are the navigator, just like our Google, your internet, no? Same thing. They will directly navigate which particular area the drug is reaching. I'm showing some other technologies, nanoemulsions, how it looks. Liposomes, just now explained, nanoparticles, nanocapsules, cationic emulsions. So here I'm using the word cationic, means positive charge. So if you give positive charge, what happens? No? PEGs generally we have used, polyethylene glycol. They will stay more time in the body. That means the drug will be available to the cancer cells more time, the cancer cells will die. That's the beauty of this one. Coming to drugs and polymers, I'm giving some examples here. You see, drugs like adenine, ampicillin, DNA, enzymes, insulin, methotrexate, primaquine, progesterone, testosterone, and polymers, different types of polymers. We are using polyisoxyl cyanoacrylate, polyisobutyl cyanoacrylate, polybutyl cyanoacrylate, polyacrylamide, polyisobutyl cyanoacrylate, albumins. Coming to characterization. So if you prepare any tablet or any nanoparticle, we should do the characterization. If you see tablet, what we'll do? You see the hardness. Normal hardness, how much? 4 to 6. If it is mouth dissolving, less than 4. If it is sustained or controlled, it is above 6. So like that, here also we should see the size, charge, how much drug is entrapment efficiency, and release profiles. And we have different methods also available. In the tablets, we'll do hardness, friability, disintegration, dissolution. So 4 characterizations will do. Here, the main important points in the nanotechnology: size, charge, entrapment efficiency, release profile. And further, we have photon correlation spectroscopy, scanning electron microscopy, gel chromatography, X-ray diffraction, DSC, that is differential scaling calorimetry, electrophoresis, hydrophobic interactions. Coming to solid nanoparticles versus liposomes. Another one, solid lipid nanoparticles. So you are making into solid because because liposomes are lipid spheres are in liquid in nature. So we want to convert it into powder form, or we can use other technology called solid lipid nanoparticles, SLN. So the main advantage, we can improve the stability. So the composition of matrix can be easily altered to tune control release characteristics, greater stability, more readily and cost effectively manufactured in large scale. I'm showing the one concept how they prepare: melting of the lipid, solubilization of the drug in the melted lipid, solidification of the drug loaded lipid in dry ice, grinding in a powder mill, dispersion of the lipid in the cold aqueous dispersion medium. Then you are going to get solid lipid nanoparticles. So nanoparticles contain up to 33 to 80% weight by weight low molecular drug, increase apparent drug solubility up to 105-fold, coating and entrapment of many types of drugs, antigens or sensors. Nanoparticles can be sterilized, filtered, and lyophilized. You know the word lyophilization? Like milk to milk powder. Okay, so people are making into powder form for the larger stability. And biocompatibility, stable in biological fluids at 37 degrees centigrade because our body temperature is like that. No neutrophil activation, no lysis, metabolized by the body, lack of toxicity and stealth properties in vivo. Coming to in vivo performance, enhanced oral delivery of drugs, transport across blood-brain barrier, in vivo targeting to solid tumors as vector for in vivo vaccines. Coming to list of drugs formulated in nanoparticles, anti-cancer drugs, nucleic acids, peptides and proteins, and others. Coming to targeting, simultaneous carrying of drug while adding targeting ligand on surface. Single chain viable fragments can be re-engineered to multimers to increase avidity to target cells. Potential tumor targets like epidermal growth factor and folate, I already told, folate receptors. Coming to nanobiotechnology, nanoparticles for diagnostic or therapeutic use, biosynthesis of nanoparticles, biological templates for nanoparticle assembly, bio-nano composites, imaging, sensing of nanoparticles or biomolecules. Coming to Taxol nanoparticles, I told you this is a cancer drug, effective against several different cancers. Taxol has been shown to be a substrate for the MDR express protein, P-glycoprotein, very low water solubility, 50-50 value by volume, chemophore, acute hypersensitivity reaction. Paclitaxel dissolved in melted emulsifying wax. Entrapment of paclitaxel would prevent binding to P-glycoprotein MDR. Coming to advantages, all ingredients are potentially biocompatible. I already told you, so compatible body is very, very important. The process can easily be adapted to include many types of drugs and excipients. So generally what happens, your drug, that is APA, active pharmaceutical ingredient, plus excipients, that is inactive. If they combine, we are getting drug product or FPP, finished pharmaceutical product. Okay, that is the concept here. So well-defined and uniform nanoparticles may be reproducibly made. No organic solvents. So here the beauty is we should not use organic solvents because they may cause again side effects in the body. The formed solid nanoparticles may have superior in vivo stability. Cell-specific ligands can easily be incorporated on the surface of the nanoparticles. Nanoparticles in chemotherapy. Stealth nanoparticles are prepared by coating them with soluble polyoxyethylene or by using dialkyl polyoxyethylene. The first cytostatic drug bound to nanoparticles was actinomycin. Second concept, chemoembolization. One of the approach, chemoembolization makes use of biodegradable particles. Administration to the liver tumor using a catheter that passes directly into an arterial tumor. The accumulation of non-stealth mitomycin C microparticles within the Kupffer cells, that is liver, has been used to target hepatic neoplasm indirectly. That is hepatic. The cancer is coming to liver. Several studies are reported for prolonged drug retention in tumor, reduction in tumor growth, and prolonged survival of tumor-bearing animals using nanoparticles loaded. So it is already proven in the animal models. Coming to delivery of anti-cancer drugs, the polyalkyl cyanoacrylate nanoparticles have been studied in recent years as a possible means of targeting drugs to specific sites in the body. Some of the anti-cancer drugs that are either entrapped or absorbed onto polyalkyl cyanoacrylate nanoparticles are doxorubicin associated with the polyisohexyl cyanoacrylate nanoparticles, because doxorubicin is the anti-cancer drug. Mitoxantrone, aclinomycin, aceclopine. So these are the drugs which are using the nanotechnology. Insulin encapsulated into nanoparticles. Encapsulation protect insulin against proteolytic enzymes. You know, we are not taking insulin by oral. Why? Because our digestive enzymes will deactivate the insulin. So that is the reason we are going for subcutaneous. Coming to nanoparticles for ocular delivery, we can use in the eye. Then we are using the brain. We are using the DNA delivery. Coming to challenges, targeted therapeutics must diffuse out of the vasculature, recognize target cells, and bind with high avidity. General regulatory considerations for nanotechnology, characterization I already told, size and charge, stability, environmental impact, develop nanotechnology expertise within the FDA and public scrutiny. Coming to multi-layer nanoparticles for drug or gene delivery in nanomedicine, self-guiding, adaptive, multi-component systems on the nanoscale for diagnostic and therapeutic prevention or treatment of disease. These smart nanomedical systems can deal with changing conditions or error correcting and can provide proper dose of therapeutic response on a cell-by-cell basis. Coming to nanomedicine, beyond the obvious application of nanotechnology to medicine, the approach is fundamentally different than conventional medicine. Nanomedicine uses nano tools like small nanoparticles that are roughly 1,000 times smaller than a cell. Nanomedicine is the treatment or repair of tissue and organ within individual targeted cells, cell by cell. Nanomedicine typically combines use of molecular biosensor to provide for feedback control of treatment and repair. Conventional cancer therapies try to cut out the bad cells like surgery, burn them out like radiation, I already explained, or chemotherapy. That is poison the bad cells faster than the good cells. That's chemotherapy. So surgery, radiation, chemotherapy. Conventional medicine removes disease cells and does not attempt to fix them. Nanomedicine attempts to make smart decisions to either remove specific cells by induced apoptosis or repair them one cell at a time. That is called regenerative medicine. Single cell treatments will be based on molecular biosensor information that controls subsequent drug delivery to that single cell. The challenge, delivery of a drug manufacturing factory, not a drug, then manufacture exactly the optimal amount for that particular cell under feedback control of upstream molecular biosensor. Nano-oncology, nano-based imaging, nanodrug delivery systems to detect small tumors, metastasis, to follow tumor progression, to follow tumor regression, to optimize drug administration, that is route, frequency, dose, avoid harmful carriers. Coming to nanotherapy, to selectively and specifically kill only tumor cells. Nano, minimize side effects. Nano products so far, micelles in clinical trials, lysosomes, and siRNA. Select nano-carriers like micelles, microemulsions, dendrimers, nanoparticles, drug polymer conjugates, liposomes like doxorubicin hydrochloride. See the how the growth is there. It started in 1771, you see, 1825, 1886, 1939, 97. You see, lot of research is going on. We are using in the nanoparticles phospholipids. I'm showing one picture for you. These are the phospholipids, very, very useful in the nanotechnology. They have head and tail. This is the liposome. Inside is hydrophilic. These are the cell membrane. So they will attach here. See, polar head group, cholesterol, fatty acids, And liposomes you see, chelation therapy, enzyme replacement, diagnostic imaging of tumors, cosmetics. So why use liposomes in drug delivery? Drug targeting main directly to systemic circulation. So kinetics, efficacy and toxicity. The drug is releasing in the body. So absorption, endocytosis, fusion, and lipid transfer. This is a mechanism. Conventional, there are different types of liposomes, long circulating, immunocationic. Liposomes help improve therapeutic index, rapid metabolism, unfavorable pharmacokinetics, low solubility, lack of stability, irritation. So custom design, lipid content, size, surface charge. These are some examples. Nanoripsin, triclosan, gene therapy, cell with non-functioning gene, adding DNA containing a functional version of the gene, then cell function normally. So we are repairing the gene. So Doxil, I told you just now, so is indicated for treatment of patients with metastatic carcinoma of the ovary who are refractory to both paclitaxel and platinum-based chemotherapy regimens and who may be refractory to topotecton. Refractory is defined as a patient having progressive disease while on treatment or within 6 months of treatment. See here, toxicity, nephrotoxicity, hematotoxicity, infusion-related effects, and ampersand B. There is there in the market. It's called ampersand B deoxyfungizone. Company called Sarabai is manufacturing. But the drug is excellent. But the disease will be cured, but patient will die because of nephrotoxicity. That means both kidneys are failed. So that is the reason we are preparing ampersand B lipid complex. That is Ablisset or Ambizome. So with this, the patient will not die and the treatment, disease will be cured. But unfortunately, it's very, very costly. The fungizone, it costs 500 rupees. It costs almost 1 lakh. So that's the main disadvantage of cost. Otherwise, no, both drugs are very, very excellent. So this is the Ambizome I told you, ampersand B liposome for injection form. It is manufactured by American company. I told you that price is a big issue. So smart nanostructures for selective drug delivery for cardiovascular disease, cancer, infectious disease, nanomedical diagnostics, targets, drug overdose, brain trauma, disease markets, especially hospitals, law enforcement. Coming to applications, pain resistant textiles, information recording layers, adaptive mask, body shell, high hardness cutting tools, chemical and biodetectors, advanced drug delivery systems. So already told you, they are using in the textiles. So they are proving in the rat how the nanoparticles are moving from one place to other place. See the colors, you can see movement of drug inside the body. And we are preparing sunscreens also with the nanotechnology. And we are using magnetic field guided drug delivery with magnetic aerosols. So they will enter into aerosol. Outside they will keep the magnet. So whatever the aerosols, it comes particular area where the disease will be cured. So these are the different nanoparticles like carbon magnetic nanoparticles. You see the size and see the role. Dendrimers, ceramic nanoparticles, hydrogen nanoparticles, liposomes, low density lipoprotein, nanoemulsions, lipospheres, nanoparticle composites, nanofill, nanospheres. These are the tumor seeking nanoparticles. So they done one experiment. So they created a tumor in the mice and they given these nanoparticles and see the size. Slowly the size is reducing. So this is a research happening in the iron-based model catalyst. So Neetha Khan, a research scientist at NETL, prepares an experiment with iron nanoparticles in NETL's Omicron analysis and surface imaging system. Research group leader Brand Boxster, standing left, and project leader Chris observed the generation and analysis of the nanoparticles. So in the lab they are doing. Nanoceuticals we are using in the atherosclerosis and using nanomedical. This layered micelle is a drug delivery nanoparticle that targets the fatty plaques of atherosclerosis. That means inside the arteries, the cholesterol will be form a clog. So this will reduce. So developing nanotechnology-based cancer therapies. Drug development issues. 1/3 of compounds in the development are water insoluble. Half fail in trials because of poor pharmacokinetics. Micelles can solubilize water insoluble compounds. Micelles can improve the pharmacokinetics. Important properties: size, charge, ratio. Most efficient polymer is about 8,000 K. Most efficient particles. Lipofectin, that is transfection reagent, is the reagent for choice for transfection of endothelial cells. It is also suitable for transfecting DNA, RNA, and oligonucleotides into mammalian cells and DNA into plant protoplasm. Coming to diagnostics, incorporate reporter molecule into nanotubes, decorate surface with appropriate targeting, couple target ignition with uncapping, detect reporter molecule in blood and breath, use in diseased animal model, use in cell lines of cancer. So coming to drug delivery system, the global market for advanced drug delivery system was more than 40 billion in 2000 and estimated to grow 231 billion by 2020 and 310 billion by 2025. That means a lot of demand is there for the drug delivery. Developments within this market are continuing at a rapid pace, especially in the area of alternative to injected macromolecules as drug formulations seek to cash in on the 4 billion worldwide market for genetically engineered protein and peptide drugs and other biological therapeutics. Nanoparticles represent interesting alternative as a drug delivery system to liposome. The application of nanoparticle is the possibility of targeting them to specific organs or tissues by absorbing and coating their surface with different substances. Nanoparticles might be superior to liposomes because many coating materials such as surfactants can lead to the disintegration of liposomes. The use of nanotechnology in medicine, more specifically drug delivery, is said to be spread rapidly. For decades, pharmaceutical sciences have been using nanoparticles to reduce toxicity and side effects of drugs. A conceptual understanding of biological responses to nanomaterials is needed to develop and apply safe nanomaterials in drug delivery. So here we should be very, very careful about nanomaterials because safety is a priority. You see, popular perception of nanotechnology, already seen a robot in 2004 and the new bread in 1995. These are the movies came on the concept of nanotechnology. Star Trek in 1987. So these are concept is nanotechnology only. Nanotechnology is taking on a life of its own, but the educational community has yet to respond, and research officials are concerned that the fledgling industry will not grow unless nanotechnology becomes a standard part of the US physics and chemistry curriculum. Challenges in science education: need for education and training in nanoscience concept to be introduced at all levels. What important concepts do elementary, secondary level students need to understand in nanoscale science and technology? Will all students have access to science teachers who are adequately prepared in this content? See, nanotech in local malls, you are getting even dresses also, fabrics also. Shoes also coming now. So physical chemical properties can change as we approach the nanoscale. Melting point of a gold nanoparticles, fluorescence of semiconductor nanocrystals. This is already told, no? Visceral leishmaniasis, I already told, it's a neglected disease. Worldwide 5 lakhs cases are there. I just now I told, no? Our ampersand B is very, very useful. Ambisome. Okay, so coming to our second, quality of life, patient treated for long periods, need for agents which are well tolerated and conventional for patients. So ambisome I told. Now coming to our second part, role of nanotechnology in personalized medicine. This is very, very important. So I will take 15 to 20 minutes. Personalized medicine takes into account individual genetic differences. Traditionally, doctors used family history, socio-economic circumstances, environmental factors. See, nowadays, no, the person is very active till 60 years or 70 years. On one sudden, the person is getting cancer. Why it is happening? Because of family history, because of socio-economic circumstances. Even nowadays, no stress also, that we are getting a lot of diabetes, hypertension. So even environmental factors also we are getting cancer. Now you see your water is polluted, your milk is polluted, your vegetables are polluted. So if you go to any doctor, no, so these things people are getting a lot of cancers. Previously, our India is called hub of diabetes. Now people are saying out of 100, 10 people are suffering with cancers. So genomic or genetic testing is very, very important. Proteomic profiling, metabolomic analysis. So if you done this, we can predict. So now what we are doing generally, we are going to doctor, it is diagnosis, selected drug. Okay, this drug is good, happy. Otherwise, the disease is not cured, switch the other drug, again switch the other drug. This is the present concept of what we are doing. Now you see, again, the screening, diagnosis, right drug, then monitoring. This is a new concept. And you see how the future concept. Again, screening, you should prevent the measures. So you should find the fault, why you are getting this, why you are getting this. If you prevent that, no, automatically, no need of taking any drug. That is a future concept. See, only monitoring. So 3 concepts are coming in our talk. So coming to personalized medicine, the ability to offer the right drug, the right patient for the right disease at the right time with the right dosage. That's the meaning of personalized medicine. Now what we are taking, everybody is taking common Dolo 650. In coming days, we cannot take, only few people as per the requirement. Genetic and metabolic data is required. We'll allow drugs to be tailored to patient subgroups. We can divide the patient into subgroups according to their genetic nature. So benefits of personalized medicine, better matching patients to drugs instead of trial and error. Now what we are doing now, doctors now, if you go to doctor, they will write these drugs. Okay, come after 1 month. He is doing trial and error only. Okay, if they are suitable, you are good. If you're not suitable, you will change to other drug. Just like we are guinea pigs. So customized pharmaceuticals may eliminate life-threatening adverse reactions, reduce costs of clinical trials by quickly identifying total failures, favorable response for particular backgrounds. So combination pill containing 2 medications for heart failure, cardiovascular disease, and or diabetes. Clinical trials did not show overall benefit across entire population. Subgroup of patients showed best overall benefit. Theranostics is a combination of the terms therapeutics and diagnostics. Combination, it's called theranostics. It is used to describe the combination of using one radioactive drug to identify, that is diagnose, and a second radioactive drug to deliver therapy to treat the major tumor and any metastatic tumors. This is the concept. You see here, this graph gives total information about personalized medicine. One side you see diagnostics, other end you see targeted delivery. You see here, so nano-carriers, like I already told, dendrimers, micelles, inorganic nanoparticles, liposomes, carbon nanotubes, metallic nanotubes. These are all comes under nano-carriers. So you are finding here. Now, once you find the disease, then either you can go for sustained release or control release or higher transport efficiency or target delivery or stimulus response delivery or co-delivery or biodelivery. So depending on the concept, you can use here. So identification of the disease one side, reduce the disease the other side. So here you see diagnostics is RX plus DX. So one side diagnostics, other side therapeutics. Unfortunately, no, we ourselves or doctors know finding the disease is a time-taking process. If the right time the doctor identifies a cancer patient in stage 1, success. But stage 3, no, it is not in their hands. So how you are going to find either PET scan or SPECT or magnetic resonance image or NIR fluorescence or ultrasound. And here you see therapeutic, the photodynamic, chemotherapy, gene therapy, radiotherapy, hypothermia. These are the different concepts. Yeah. So what is PET scan? So positron emission tomography. SPECT means single photon emission computed tomography. So nanostructures which are of a suitable size scale to interact directly with many biological structures and systems are going to be vital tool for physicians to gain better information about patients' bodies and will allow them to work directly with the body to prevent and cure diseases. A major focus of nanomedicine research to date has been novel methods of targeted drug delivery using nanocapsules and functionalized nanoparticles. Nanoformulations of existing drugs can enable simpler administration, increase safety, or increase efficacy. There has also been a great deal of research interest in nano-enhanced diagnostic systems. Nanoparticles and other nanostructured devices have been found to be more effective at detecting or quantifying disease markers than more conventional counterparts, particularly when looking for very small quantities of a chemical. Antimicrobial surfaces and products using nanoparticles. We are using silver nanoparticles also. Personalized medicine is a huge movement in the modern medical world. It aims to move away from the traditional practice of prescribing standard doses of standard drugs for a condition to every patient and shift the focus on to targeting the precise drug and dose required according to the patient's physiology. That is a concept of personalized medicine. So this is achieved by detecting and tracking molecular biomarkers, which indicate the presence and level of activity of a particular biological system in patient's body, whether inherent or foreign. Another measure is particularly important in cancer treatment, where the chemotherapy drugs used can be very damaging to healthy cells as well as cancerous ones. And the exact genetics of the tumor cells can vary widely between patients and even between locations in one patient's body. Part of the emerging field of personalized medicine is pharmacogenomics, analyzing the genetic makeup of the patient to determine whether a particular medication will be successful or if it will have any adverse effects. Nanotechnology solution for personalized medicine. Most of the recent developments in personalized medicine have been directed at better diagnosis and treatment of cancer. Cancer is one of the biggest killers, and existing treatments can usually only improve the patient's chances, and outright cure is very rare. Personalization of treatment of cancer is a promising way to increase our ability to fight cancer, however, without necessarily requiring discovery of a revolutionary breakthrough drug. By determining which drug is particular patient will respond best to and correct the dosage for. There are several ways in which nanotechnology can help with these developments, primarily by making the diagnostic process simpler, quicker, or less invasive by requiring less tissue. Genetic analysis is a crucial part of personalized medicine, particularly in cancer treatments, where the genetic signals can be so varied. Techniques are being developed for high throughput DNA sequencing using nanopores to obtain genetic information from a patient so that target medication can be selected. The unique electronic and mechanical properties of nanostructured carbon materials like carbon nanotubes and graphene, very interesting as the active components of nanosensors, which can detect incredibly small concentration of a biomarker, in some cases down to little as few molecules. Many of these technologies can be combined into a lab on a chip, a portal diagnostic device which will be able to run tests rapidly and without requiring a large sample of tissue or blood. These devices will be able to be used in general practitioners' offices or in remote locations, which will make a huge difference to the speed at which a definite diagnosis can be given, a crucial step for cancer patients, where their chances can be depend very strongly upon how early treatment starts. I don't know, because if the treatment starts in stage 1, 75% chance of the success. But unfortunately, the patient is unable to identify himself, and that we are not going to doctors. So naturally, doctors, or we cannot blame doctors because we are going late. So that time they have only one chance is chemotherapy or radiation or surgery. But third stage, very difficult. Only chemotherapy is the option. So this sort of technology will also have a huge impact as the concept of personalized medicine spreads to include the treatment of a broader range of disease. A commercial lab on chip capable of diagnosing diseases rapidly and providing the physician with enough information about the patient physiology to select the correct drugs and dosages would transform the world of medicine completely. Graphene nanopores can be used to sequence DNA. The DNA can pass through the pores, preventing the flow of ions through the pore, resulting in a characteristic electrical signal. Personalized medicine means patient's tissue-specific targeting of disease, variability of each individual has to be considered before developing a suitable treatment strategy. Diagnostics is inevitable for early diagnosis and therapeutic applications for screening of patients depending on the responses developed by the patients toward the therapy. Patients with better results and favorable outcome can be continued with the same therapy, while the patients with less response can be treated with some other therapy. It will increase the safety profile because individual specific protocols will maximize the benefits and chances of treatment. Personalized medicine can be very successful in the case of treatment or control of cancers. Most of the anti-cancer therapeutics fall into three major modes of actions. First one, those molecules having the ability to specifically binding to a specific biomolecular target or molecular product obtained from altered gene expression or pathways and are key for differentiation of cancer cells from normal cells and killing of them. Second strategy includes supporting host immune response in removal of or killing of tumor malignant cells, and third by increasing the permeation ability of drugs or anti-cancer agents. Anti-cancer agents can work by invading the cancer cells or by attaching to the surface proteins present on the cancer cells. Most of the small chemical agents mainly attack cellular process related to growth or division such as DNA replicase, tubulin polymerization, and enzyme catalyzed biochemical reactions. In the invasive mode of actions, there is also use of small nucleic acid sequence-based hybridization, where the sequence can bind to the complementary sequence or to the gene product. Receptor-based therapies involve binding of the anti-cancer agents to the cellular surface proteins. Small chemical entities for the treatment of the cancer have seen tremendous growth between the approval of various agents with the similar functions. The imatinib, the cryogeninib, viminafenib were used to treat patients suffering from cancers. No, monoclonal antibodies. These drugs comes under monoclonal antibodies. So coming to human variation in human beings, 99.9% bases are same. Remaining 0.1% makes a person unique. This variation can be harmless, harmful, or latent. So SNPs means single nucleotide polymorphisms, single base change in DNA. SNPs arise as a consequence of mistakes during normal DNA replication. So by birth, these mistakes are coming. Because of that, the people are getting this type of diseases, either cancer, diabetes. I don't know, because of family history. So always doctors would, the first question they will ask, what is the family history? Why they're asking? Because any genetic variation is there or not. Pharmacogenetics, you see here, most people metabolize the drug quickly. Doses need to be high enough to treat leukemia and prevent relapses. Others metabolize the drug slowly and need lower doses to avoid toxic side effects. A small portion of people metabolize the drug so poorly that its effects can be fatal. Okay? Very few people will get this type of toxic effects. So kinetics, what the body does to the drug. Dynamics, what the drug does to the body. You already studied in your B Pharm first year. So I'm giving you an example here, protein metabolism. 80% protein normally converted to glucuronide eliminated by kidney. Very good. 80% people, no issue. Only 10 to 20%, what happened? No, the protein is metabolized into morphine by CYP2D6. Inhibition of CYP3A4D or rapid metabolic variants of CYP2D6 during renal failure would show toxicity. The 20% drug is staying in the body, your kidneys are going to fail. So because of variation in the enzymes. So that's I'm showing here. So there is in the lab they prepared AMPLICHIP CYP450 range of drug metabolism phenotypes is observed for individuals based upon the cytochrome P450 genes. So we should find the risks, known side effects, medication errors, product quality, defects. So preventable and adverse events. So if you take like what happens, no, there is a possibility of injury or death. Liptar, you know, very famous drug, blocks the production of cholesterol in the body, may reduce risk of hardening of the arteries, which can lead to heart attacks, stroke, and peripheral vascular diseases. But no, nowadays no, so many companies are calling recall, means taking back, because product defect, delivery system may be possible. I'm giving here percentage, chances, packaging, fill problem, contamination, sterility, sometimes no impurities are present. Because of that impurities, no, again, the drug becoming prone to cancer, and formulation or substitution, adverse drug reports, and labeling. So these are the issues are coming. The companies are recalling their own products. Coming to in silico drug discovery, need 3D structure, scan a virtual library for a small molecules and dock them to a site of interest on a protein structure, predict binding energy, filters thousands of compounds relatively quickly. When you are studying human genetics, you are studying the information that goes into the making of man and how that information flows from one generation to next generation. To be able to do that well, you have to know the population structure. We can basically take the list that includes everyone in the country or 2,000 people with schizophrenia. We can know within minutes exactly how everyone is related to everyone else, which is key for being able to study the genetics of anything in a sensible manner. So does my doctor know enough about genomic medicine to be advising me? Are there genetic counselors available? Will the test only be for this condition? What if I am susceptible to another disease? Who will know about this? Doctors or insurance companies? How exactly will the results be kept secure and in confidence? That's a big issue now. If tomorrow genetic data is coming to doctor or insurance agents, so naturally what happened? No, the insurance companies will not give to insurance. They will charge more. Why? Because they already knew the thing. So there's a big issue now. Ethical issues coming now. So more drugs may succeed in clinical trials due to positive outcome for smaller subset. Will public health cover the cost of genetic testing? How will insurance premiums be affected? So naturally, what happens now? These insurance premiums will be doubled or tripled or 10 times. I don't know. It's unimaginable. So science still early, limited data in public domain, many variations not clinically significant, expensive to test for genotype, ethical issues in testing individual genotype, unclear how to deliver information to the practitioner. So finally, I'm coming to conclusion. The concept of nano formulations, nanotechnology is capable of providing medical professionals with a vastly greater set of diagnostic tools with less invasive procedures and more rapid results. This will help to use the new age of personalized medicine, where all the additional data about the patient can be leveraged to tune the treatment to their precise needs. This will have a particularly strong impact in cancer treatment, where the chemotherapy drugs, which will have the least adverse effects on the patient, can be selected. Drug delivery system using functionalized nanoparticles can also be used in tandem with the diagnostic techniques to improve targeting of drugs to particular parts of the body. All aspects of nanomedicine, there is a complex regulatory minifield to navigate before these technologies can be see public use. Target drug delivery system must be shown to be safe and reliable enough, and diagnostic systems must demonstrate a sufficient degree of accuracy. However, the benefits available from exploitation of this technology should drive the process to move forwards, and new research is emerging all the time, which strengthens the case for nanotechnology in medicine. So thank you very much for your patient listening.
Thank you, Dr. Prabhakar Reddy. Thank you very much. You are giving us speak in a very elaborated way. Hope everyone might understand very well. So we have a small 3-4 questions here. I'll just read. You answer them, Mr. Prabhakar Reddy.
Sure.
When was the nanotechnology first used in medicine?
1990, no, liposomes, 1995. Ambisome came into picture.
Okay. How can we use nanotechnology in our daily life?
Now I already told, no, in daily life, no, medicine I already told, but you are seeing your mobile phones. Previously you've seen your computers.
Hmm.
Even in coming days, we will see nano dresses also coming, even nano shoes also coming, and sunscreen agents are coming. Always there in the market, but unfortunately, it is not came in the Indian market. It is there in the Western market.
Okay. What type of nanoparticles are used in most of the cancer treatments? Most of the cancer treatments.
Again, no, we cannot say direct because in cancer also there are different varieties of cancers. I already given nanomaterials. So
Okay.
each one, according to their requirement, they can use.
Which is the best method in nanotechnology to use in transdermal patches?
But nanotechnology still now, no, they are not using in transdermal technology.
Transdermal patches is not
no,
there.
No, patches not can. Only sunscreen,
Lotions.
topical lotions came. Patches not can.
Lotions and aerosols.
Yes.
What are the major and minor challenges facing in nanotechnology?
Cost is a big area now.
Okay, that is major. Minor?
Major. Minor, no. Funding is a big issue now.
Finance.
Finance.
And this is.
Infrastructure main cost.
Okay. How can we estimate the drug concentration at the tumor site?
We cannot estimate the tumor site. Only we will take the blood from the body. I don't know kinetic studies. Different time points. We will take the blood. We will check it. How much is there?
What are the risks of nanotechnology in medicines?
I don't know nanomaterials because these nanomaterials sometimes they are causing a lot of side effects. Only that is a worry. So that is the reason.
Major side effects are
materials, no, biocompatible, biodegradable. We should think those things. If they are not coming from body, no, kidney failure is coming.
Okay. These are the major risks.
Yes.
Can nanoparticles changes the DNA?
No.
It cannot.
No. No drug will change the DNA. It is.
Okay. Do nanotherapies available in all the hospitals in India?
No.
Only in particular.
Only major hospitals.
Only major hospitals.
Yes. Because I told no, because a lot of infrastructure is required, trained person is required.
Okay.
And all people are not able to pay that amount.
Is chemotherapy is personalized? Based on
Chemotherapy?
Chemotherapy is personalized to patient?
Yes, natural, no?
It is.
Yes. Because we don't know which person, no, sometimes we are giving a lot of drugs. The drugs may not be suitable. Immediately doctor will change to other drugs.
Okay, Dr. Prabhakar Reddy, these are the questions we have got into the chat box.
Thank you.
I request anybody else can ask me anything. So he'll answer. Anyway, any other questions?
NASA ji here.
I can simply say the presentation given by Prabhakar Reddy is excellent one.
Yes, it's true.
He has covered all the aspects of nanotechnology, personalized medicines, side effects, how the nanotechnology is useful in different conditions, particularly cancer and related issues. Presentation is excellent one. Thank you very much.
Thank you, NASA ji. Thank you very much.
Thank you. Shall we go for the next session? The Professor Garu?
Yeah, please.
May I request Mr. Gampa to share the vote of thanks?
Yeah.
And a small issue, and after the vote of thanks, we will have the national anthem.
Good afternoon. I, Professor Vijay Kumar, Professor, Department of Pharmacy, KGR Institute of Technology and Management, happy to propose vote of thanks. I personally thank our national president, mentor, towering personality in the field of regulatory division, Dr. Atul Nasagaru, for giving his opening remarks of today's webinar. I thank our state president, Mr. Koteshwar Rao Garu, for his support in all aspects to elevate IPGA-Telangana state branch at national level. Is always young and energetic and a great visionary. He is the main person to lead IPGA-Telangana state branch to a national level. I thank today's chairman of the webinar, CH Kashi Vishwanatha Garu, for coordinating the today's event in very excellent manner. I thank especially Sri Nishit Garu for technical support. I thank today's Veera Reddy Garu for his elaborated talk on role of nanotechnology in personalized medicine, which is highly useful for budding pharmacists and young entrepreneurs. Last but not least, I thank Indian Pharmacy Graduates Association Telangana State life members for their continuous support for their continuous support to strengthen the pharma profession under the leadership of Koteshwar Rao Garu. I also thank today's Educational Institute, web partners, management and director of Department of Pharmacy, KGR Institute of Technology and Management, Hyderabad. I thank you one and all.
The link for e-certificate is shared. Members may download them from the link given in the chat box. Over to Nishit.
Shall we go for national anthem, sir?
Yeah.
Please, sir.
Yeah, Nishit, it is, I think, national anthem.
जनगणमन अधिनायक जय भारत भाग्य विधाता पंजाब सिंध गुजरात मराठा द्राविड उत्कल बंगा विंध्य हिमाचल यमुना गंगा उच्चल जलधि तरंगा तव शुभ नामे जागे तव शुभ आशीष मांगे गाहे तव जय गाथा जन गण मंगलदायक जय हे भारत भाग्य विधाता जय हे जय हे जय हे जय 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 हे
बोलो भारत माता की
Jai.
Hey,
One submission. If anybody observed any problem for downloading certificate, please WhatsApp me, I will send.
You put your cell number there. Dr. Vijay Kumar, you can message the cell number.
in the chat box.
Yeah, in chat box.
It is only for today's participants.
Okay.
Thank you, Prabhakar Reddy, once again.
Yeah.
It's wonderful.
Thank you. Yeah, thank you very.
You have covered almost all the aspects. Just there are questions, I have read it. Otherwise, I also knew and I also got the answers for these questions. But
Very
as some people are asked, I have brought to you.
Thank you very much, sir. Thank you. Special thanks to Nishit for support, continuous support.
Yes,
He's suffering with fever. Still, he support. Really, sir. Thank you very much, sir, Nishit, sir.
Okay, sir. Always welcome.
Yeah. Thank you. Thank you,
Sir,
Wonder.
can we conclude now, sir?
Yeah, we can, sir.
Yeah.
Okay.
You can close, sir. Thank you, sir. Nasaji, thank
Thank you,
Thank
Dr.
you very
Mukherjee.
much. Kodeshwara sir, thank you very much. Vijay, thank you very much. Yeah, all the participants, thank you very much for your patient listening. No participant will left till now. Great.
Yes, it's true. Still
We
there
should
are.
appreciate- Still they are. Generally, they will leave at the end of the talk, but really appreciate all of you.
No, but your the way of explanation is also very amazing. So that's the reason people still they wanted to listen some more.
Thank you very much. Generally, we'll see in the physical classes. Now I'm seeing in the webinars also. Really appreciate all of you.
Yes.
Great.
Thank you once
So this-
again for you from your IPGA TS.
Yes. Special thanks to Telangana IPGA
Yes.
because they given a opportunity to talk to all of you.
Yes.
Thank you one and all.
And then we can plan every month once.
Yeah, definite, sir. Definite. Monthly ones.
Monthly once we'll do.
Yes.